Thank you once again for joining me on behalf of the Antibody Society. My name is Jess, and this is another episode of The Researcher Behind the Image for our 2024 calendar. Today I'll be joined by Gabriel, who is a PhD student looking into 3D organoids of the brain. So let's hear all about his research behind the April image.
So I'm basically investigating what is the human cortical interneuron cell death in using two approaches, a xenotransplantation model and a neural organoid system. So I can explain you a bit more about this.
Great.
So we know that from mice, we know that in the cerebral cortex, we have like two main groups of neurons. So we have excitatory neurons or pyramidal cells, and inhibitory neurons, also called interneurons. So from mice, we know that interneurons are generated in the ventral part of the brain. So this region here of the brain, and then these are generated during embryonic development, and then they have to migrate all the way from the ventral part of the brain to the cortex. So this is this region here above. And but the interesting thing is that we know that interneurons are generated in excessive numbers during development. So after they reach the cortex, they have to like adjust their numbers to the proper final numbers. Why is this important? Because interneurons control the activity of the other neurons, the excitatory neurons. And why is this important again? Because like when there's too much activity, this can lead to different disorders, for example, to epilepsy, schizophrenia. So it is important to have like the proper numbers of interneurons and excitatory neurons in the cortex. So as I said before, from mice, we know that interneurons are produced in excessive numbers during embryonic development, and then they adjust to their final numbers just after the mice are born. So between the day 5 after mice are born and the day 10, there's a massive cell death occurring in these interneurons. So around 30% of the interneurons that were born die after the mice are born. And
That's
so this
really
is how
fast.
yes, and this happens only in 5 days. So this adjustment is important. Otherwise, if this didn't happen, then we would have like too many interneurons, and maybe like the balance, this excitation and inhibition balance that I've been talking about, like could not be reached, and this, as I said, this could be leading to different disorders. So we know this from mice.
Out of interest, do they know this window in humans?
Exactly, that's what I'm trying to.
You're getting there. So one step ahead of you. Keep going.
This is what I'm trying to answer with my question. So, because interestingly, in humans, we know that there's 3 times more the proportion of interneurons than in mice. So, what would this mean? So, would this mean like maybe there's no cell death of interneurons? That maybe like all the interneurons that are born in humans, they are like kept during like the entire life, or what's going on here? So, and also like as we don't see like there's more inhibition in humans, although there's more interneurons proportionally. So what is happening here? So this is basically what I'm trying to answer with my question, is like, how is the final number of human cortical interneurons regulated? So let me just to next slide. So we know that in humans, interneurons are also born in the ventral part of the brain, and then they migrate towards the cortex. But it would be ideal to study this process in humans, but there's some disadvantages. I mean, like it's not like
Not an easy task.
It's not like we can experiment with humans, or also it's like very difficult to have access to like a lot of human tissue to be able to do these experiments. So this is where brain organoids come. So basically, what is a brain organoid? So you take pluripotent stem cells, that are these cells that have the capacity differentiate into different types of cells. And you basically plate them in ultra low attachment plates. What is happening then? So when you plate them in these ultra low attachment plates, these pluripotent stem cells tend to self-aggregate, and then they form the three layers of embryonic development. So they form the mesoderm, the endoderm, and the ectoderm.
Okay.
And
Are
then
these
we
proportional amounts out of or does it have a preference to one type of layer being more prevalent than another?
I actually am not sure about this, but I think like the main layer that is formed is the neuroectoderm.
Okay, cool.
And so we know that ectoderm is the layer that gives rise to all the nervous system. So sometimes we can push like more the formation of this layer in these cell aggregates that then can that are called embryoid bodies.
Yeah.
So if we push more the formation of this layer, we can get a better neuronal induction. So after we push these embryoid bodies towards a neural induction,
Yeah.
we can further specify these cell aggregates to either form a more dorsal part of the brain or a more ventral part of the brain. So as I mentioned before, so why would this be important? So because we know that in the ventral forebrain is where interneurons are generated, and in the dorsal forebrain is their final destination. So if we want to generate, for example, interneurons, it would be better to generate ventral forebrains, ventral forebrain organoids where these interneurons would be generated.
Yeah, that's amazing that you can make it so specific to manipulate it to be one region.
Yes, actually, like you can push organoids to generate different regions of the brain, and this is just by adding like patterning molecule that we already know, like from studies from mice.
Yeah, no, amazing.
So this is why we use organoids, and so organoids also like present an advantage over to the cultures. So, for example, in organoids, we can recapitulate better the structure of a particular organ compared to the cultures. Also, the cellular composition is better in organoids. It's more similar to the endogenous composition in tissue.
No, definitely. And I noticed, so the image that got selected for our calendar, your organoid that you used there was 35 days. So how long does these usually last for? How long does it take before they're usable for experiments?
So basically, you can keep organoids. I think like there's some groups that have kept organoids up to 7 years. So you keep
was
them,
not
like
expecting that.
yeah, for a very long time. And so in my experiments, because I don't know exactly when to expect interneuron cell death if it's happening in human cells, I would need to keep my organoids also for a long time, at least a year, to see whether this process is happening. Basically, this is the approach that I'm going to follow. So I'm going to use a I'm going to have like two different cell lines. One of them is going to it has a report that tells me when a cell becomes an interneuron. And then the other one, also when a cell becomes an interneuron, but also when this cell is going to have a deletion of the Bax gene, which is a essential for apoptosis. So basically, these cells will not go through cell death. So it's basically to have a control, a control cell line. So we would expect the wild type cells to go through cell death and the mutant cells to not go through cell death.
Oh, brilliant. Yeah, that's a really neat system. Is that one that your lab has come up with themselves?
Yes. This is something that we have designed just for my PhD project. So this is something that I've done all myself.
Oh, great.
And so the idea is basically to generate ventral organoids. As I mentioned before, these are the organoids when interneurons are generated. And one with the mutant cell line and one with the wild type cell line. And then just basically isolate the interneurons, the wild type and the control interneurons, and transplant them into the mouse cortex in equal proportions, or culture them in a dorsal organoid. So a dorsal organoid basically recapitulates the cortex of the cortical part of the brain. So, and this is where interneurons, the final destination of cortical interneurons. So we would in theory need like ventral organoids to generate then the interneurons, and a dorsal organoid to transplant the interneurons too.
Okay. Will this allow you to alter those proportions? So even though it's a mouse model, could you have more of those interneurons to better appropriate what's present in the human? Because you said there was like 3 times the amount in human.
Yes, exactly. That's also what we could manipulate with this approach.
Brilliant.
And so basically after I have done these transplantations, so it's just wait, no, and then
just pain.
Yeah, just wait and then do a survival analysis of these two groups of cells and see whether like at a specific time point I can see a higher proportion of the mutant cells, which then would mean then that the wild type cells, so the interneurons that were not Bax mutants, would be going through cell death, and then by that I will be able to identify when cell death is happening in human cortical interneurons. If it's happening again, because we don't know, we were not sure if this process is actually happening in human cells.
Yeah, no, very interesting.
So here I have I think this was a day 45 dorsal organoid. So again, the one that recapitulates the cortex, and I'm showing a staining for 3 different markers. No, so well, DAPI just staining for the nuclei, and then MAP2. MAP2 is a pan-neuronal marker, so it's basically showing us where the neurons are located. So as you can see, in organoids we can see these what we call rosettes that are structures that resemble the ventricles of the brain. And this is where cells are generated. So this is where the progenitor cells are located in these kind of ventricle-like structures.
So are they PAX6 positive then, looking at that usually?
Exactly. These are PAX6 positive. We expect these regions to be PAX6 positive. So
PAX6. I think those were the 2 markers you used for the calendar as well, MAP2 and PAX6. So that was well selected. Thank you.
Exactly. So PAX6 is a marker of dorsal progenitor. So it's the one is the progenitor that gives rise to neurons in the cortex. So we can see that all the ventricle-like structures are PAX6 positive. So meaning that these organoids are have actually a dorsal identity, which is good. And then so I'm also showing TBR1. So TBR1 is a marker of deep layer neurons. So for example, layer 5 and 6 of the cortex. So if we see the expression of this marker, then it means that it's a cortical neuron.
That makes sense, yeah. And are these all antibody-based fluorophores? Well, except for DAPI?
Yes, all of these are antibody-based fluorophores.
Brilliant.
All what I'm showing are antibody-based, fluorescence-based antibodies.
Staining, yeah. No, they're absolutely gorgeous. So I see that your approximate size is 100 micrometers. Is this a usual size of one of your organoids? I'm guessing it's around, what, 500-600 micrometers in diameter?
Yeah.
Actually.
It depends on the age. So they get a bit bigger while they become older. I think like they can reach like their maximum size around 50 days old.
Yeah.
And so I think at that time they can be around 4 or 5 millimeters of diameter. No, so you can see them. So, yeah, so you can see them. You can also see them grow, how they can become bigger. So yeah, they become like your children.
Well, after that investment in time, not surprised there.
Exactly.
What microscope are you using for these images too, by the way? Because they're gorgeous, lots of detail.
Yeah.
So it was confocal?
Yes, it was confocal. This was confocal imaging.
Oh, great preparation then. They look beautiful.
Thank you. I can also show you some. So these are, as I mentioned, these are dorsal organisms, but I can also show you of my ventral organisms where I generate the interneurons and from which I.
Brilliant. Yeah. So you'll have to explain how the markers differ for us, so we get a bit of an understanding. Oh yeah. They do look physiologically different slightly.
A bit, yes, a bit.
Yeah, they're a bit different.
So this is a ventral forebrain organoid. This is a day 35.
Yes.
So I'm just showing the nuclei with API, and then I'm showing NKX 2.1. So NKX 2.1 is a marker of a ventral region of the forebrain that generates interneurons.
Okay.
And this is mainly in progenitors. So we'll expect this marker to be present in the ventricle-like structures again. And
Yes.
you can see you can see it enriched in the ventricle-like structures. Although you can also see some expression in where neurons are located. So in all the regions that are not SOX2 positive. So SOX2 is a marker of progenitors. So this is marking all the progenitor cells. So you expect this to have to be present in all the ventricle-like structures.
Okay. Yes.
And NKX 2.1 just in the ones that are actually acquired a ventral identity.
Oh, okay. So what if it's double positive for those things? They're just not as far along in their development then, if they're SOX2 positive and NKX 2.1 positive?
Actually, that's what you would like to have, to have double positive, NKX 2.1 and SOX2 positive.
Yep.
Because that would mean that those are progenitors that are properly patterned towards a ventral identity.
Okay, brilliant. You do have a lot of those, it looks like.
Yeah. And also, but you can see that there's some parts of the ventricles that are not NKX 2.1 positive. So there's also there's also some in the ventral forebrain, there's also some progenitors that are not NKX 2.1 positive. So this is not a surprise because there's also other regions in the ventral forebrain. So this is expected. So it doesn't mean that it's not properly patterned. It's just maybe it's a region of the ventral forebrain that is not our region of
cells.
Yeah. And then I'm finding.
Oh yeah, you're going to go to LHX6 then?
Yes, so LHX6 is basically the marker that we're more interested in because this is a marker of interneurons, of postmitotic interneurons. So basically, after progenitors become a neuron, as you know, once progenitors commit to a neuron, so they stop going through cell cycle. So they don't go through cell cycle again, so they don't replicate anymore.
They're arrested in that state, yeah.
So they become postmitotic. So this is when LHX6 is expressed. So in ventral forebrain interneurons. So this would mean when we see expression of LHX6, this is a postmitotic interneuron, and that would be our cell of interest.
Okay. No, that's really interesting that they follow quite closely the edge of the high SOX2 expression. Exactly. They're right on the edge.
Exactly, and that's what we will expecting. So we will be expecting LHX6 to be expressed in SOX2 positive regions, because these are not LHX6 positive cells are not supposed to be progenitors anymore, and so we expect them to be everywhere else, but in the ventricle-like structures.
Lovely. So how are you moving forward with this research then? How are you looking at apoptosis? Well, it was apoptosis, or what type of cell death are you looking for?
Yes, exactly. I'm looking at apoptosis. So as I mentioned before, I am generating a cell line that wants to go through apoptosis because we're deleting the VAX gene, which is critical for apoptosis, one of the main pro-apoptotic proteins.
Yes.
So basically, what I'm doing is after I've done the transplantations of the wild-type cells, wild-type interneurons, and the mutant interneurons. So I will know what number of cells I'm transplanting into either the mouse cortex or a dorsal organoid.
Yes.
And then after a certain time, what I'm going to do is just count the number of these two cell populations to see if there's a decrease in the proportion of the wild-type cells. And then this would tell me that there's cell death happening. And so it's basically just cell counting after this. After I've done the transplantation, it's just going to be cell counting.
And what stages will you do these? Because I'm guessing that will be indicated from how your preliminary work that you've shown us has gone. Is about 35 days what you're aiming for in terms of when you're going to count, or earlier, later?
I'm going to count at different times. So the idea is to count, I think, maybe 2 weeks after transplantation, 1 month, 3 months, 6 months, 9 months, and up to a year. Because as I mentioned before, I don't know when this process is happening in humans. And so it's just basically trying to identify the window where this could be happening.
Yeah, brilliant. It sounds like an excellent project and you're well on your way. Did you have any other parts of the presentation that you wanted to go through as part of this? Or?
Let me see if there's anything. I have just done my first preliminary transplantations, but I don't think I have them here.
You don't want to give away too much as well. So. But that's brilliant. I can portray that. So if you stop sharing the screen and just, I think you've covered most of what I think our viewers will be looking for. So I will just change to gallery.
Thank you to all of you, especially our members, for joining us again here today for our Research Behind the Image edition of April. A huge thank you to Gabriel once again for taking us through his fascinating research, and all the best of luck with the rest of your PhD in the future. Now, hopefully we'll be seeing you all again soon for our next edition. Thanks!